Hello, I'm your host, Ray Dogum, and welcome to VibeCast. Thank you for joining us as we explore the exciting advancements in technology-enabled collaboration to excel important drug development. VibeBio seeks to find every cure for every community. We see a future where communities of biopharma experts and patients collaborate to identify high-potential medicines and have the ability to access capital to actually develop them. The VibeCast is our weekly informational podcast where we explore some of the hottest topics in drug development and technology innovation with some of the dynamic people that make up the Vibe community. Join us to learn, imagine, question, and help us identify and develop solutions together. Our guest today is Michelle Werner, a seasoned pharmaceutical executive with more than 20 years in the industry spanning both commercial and R&D. Among many things, Michelle is the CEO of Alltrna and on the board of a nonprofit called Rare Disease Renegades. Michelle, welcome to the show today. How are you?
It's great to be here, Ray. It's a pleasure. Thank you so much for having me. I'm excited for this discussion.
Yeah, I'm excited as well. Maybe we can get started with you sort of telling the audience a little bit more about yourself and sort of your career path that led you here.
Sure. So as you mentioned, I'm the CEO of Alltrna. I'm also a CEO partner at Flagship Pioneering, and I've been here for about a year and a half in this role. So it's really exciting times. I've been in the industry for well over 2 decades. I'd say predominantly in large pharma, where I've been for the last 20 years. And I've had the opportunity, or really I'd say the privilege, to be able to work on a number of different product launches throughout that time, mostly in the oncology space across 3 different companies: Bristol-Myers Squibb, AstraZeneca, and Novartis, in a variety of different roles: sales, marketing, market access, general management, large kind of global strategic long-term roles, but also in-country operational roles in a variety of different places. And, I feel like when I look back on my career, I've been able to make play a small role, I'd say, in the advancement in oncology that we've seen over the last 2 decades, which has been very humbling and rewarding. And, it was only recently that I decided that I wanted to transition my career into the biotech field. I actually started my career off in biotech. And so to me, it's a bit of a return to the biotech sector, especially here in Cambridge, Massachusetts. And really looking for something that's a bit more fast-paced and dynamic, very close to science, very close to patients, feeling like I can have a real impact every single day. And that's something that's really drawn me here.
That's amazing. And thanks for sharing that background. And something that I, read about you while doing some research is that you're also, you're coming at this from a personal level too. So you have a child with Duchenne muscular dystrophy, and you want to talk a little bit about that and how that influenced sort of your passion?
Sure. Yes, I'd love to. I mean, so that's correct. I'm a mom, like many executives. I have 3 kids. Right now, they're between the ages of almost 12 and 16. And my son, who's 13 years old right now, his name is Caffrey, he was diagnosed with Duchenne muscular dystrophy in May of 2020. So right at the sort of height of everybody locking down in the pandemic is when we were going through this, I guess, the sort of culmination of a diagnostic odyssey that is quite common in the rare disease setting. And we got the official diagnosis. He was 10 years old at the time. And certainly, for myself, who's been in the industry for a number of different years, that has had the opportunity to work on a tremendous number of advancements during my career, of course, one of the first things that I do is, when you're faced with a, seemingly catastrophic diagnosis that DMD can be, is, well, what's the latest research? What are the clinical trials that are ongoing? Who are the key experts in the field that we should be seeking care from? And yes, there are key experts, and that's all well and good. And certainly there are clinical trials that are underway. But it was very surprising to me to see how difficult it was to get information, how fragmented the space of rare disease can be. And unfortunately, at least in our circumstances, how little innovation there was, and how the standard of care just hasn't really changed very much for DMD over the course of the past 10 years. Now that may be changing, but when I do that search on clinicaltrials.gov and there's absolutely zero clinical trials that he's eligible for, it's really disheartening. And that is, was certainly a major inspiration for me to think differently about my career. Now feeling like I've had at least the opportunity to play a small role and make an impact in oncology, if I can look at the next 20 years and feel like I can have a similar impact in the rare disease setting, this is something that really drives my passion every single day, because I do think that oftentimes these are patient populations, certainly that are underserved, that are overlooked by major pharma companies, such as the ones that I've worked for before in the past. And I think there's a lot more that we can and we should be doing. And that's why I'm here.
Absolutely. And that's something I hear a lot from families with, children with rare diseases, that they think that there is things that they could be doing. And so they start a community or a patient organization. And what you started, Rare Disease Renegades. Do you want to talk a little bit about that and how you founded it? And I think, first of all, thank you for doing that. And it's hard to be able to juggle all these different jobs, including being on the board of this organization. So, but I'm thanking you from all the other families that are affected by this as well.
Well,
So
thank you so much, Ray. And what I will say is, I think that there's actually some phenomenal patient advocacy groups that are in existence across a number of different diseases, including those that focus in Duchenne muscular dystrophy. What I think is actually also something that we noticed a lot during our experience, at least, is how, like I said, there's information that's available, especially when it comes to understanding the disease. But what I found to be a little bit more disjointed is that there are like pockets of like really good ideas and research and funding that are happening in all kinds of different places. But the ability for them to really come together or, seems to be not so straightforward. Right. And to me, I definitely believe in this principle of, if we work together and if we collaborate on great ideas, then this is where a lot of the inspiration is going to come from for truly groundbreaking innovations. And we need to figure out those models that allow different groups with different ideas to, get together and to really be able to share that and try to tackle the same problem, but instead of tackling them in sort of silos separately, to try and tackle them together and bring that collective brain power into one setting. I mean, this seems like, something that could be very worthwhile. And that's really the idea behind Rare Disease Renegades. Like, how can we create these opportunities or this ecosystem that brings some of these components together and help bridge some of those gaps that exist in the rare disease space today?
yeah, oftentimes it's those nuanced symptoms or those nuanced discoveries that only that one patient might have, but can lead to some very interesting discoveries. So I appreciate that.
Yeah.
Let's switch gears a little bit and talk about your experience at Flagship Pioneering and just what is Flagship Pioneering? What have they been doing? And what is your role there? And what would you like to achieve while you are
Yeah.
there?
So Flagship Pioneering is really an incredible organization that's been around for a while. And is really all about the venture creation company for a number of different bio platforms. I can't remember the numbers of companies that Pioneering has now inspired off the top of my head, but there's dozens of them at this point in time. Perhaps one of the most well-known or famous at this stage is probably Moderna, which is a company that many of us know because of the important role that Moderna played in bringing forward mRNA vaccines for COVID-19. But there are a tremendous amount of other, numbers of other companies that are all really trying to tackle these very big scientific questions, which I find to be truly inspirational. So I do have a dual role. I'm really predominantly the CEO of Alltrna, but I'm also a CEO partner at Flagship Pioneering. And really what that means is, I take, my experience that I've, built up over the course of my career and not only throw it into being able to help lead Alltrna to be a successful company and grow Alltrna to be a successful company, but also play a role in inspiring some of the other flagship companies from also for them to also be successful and to contribute to the greater good across the Flagship Pioneering ecosystem. So that's a little bit about my roles there. And honestly, my experience here has been really tremendous. The last year and a half, I feel, have been really rewarding and very liberating. I mean, not only is it super exciting to day in and day out be working with the team here at Alltrna on some really exciting work, which I'm sure we'll get to in a little bit, but I also find the environment where to be incredibly inspiring, because this is truly an environment, and this is really what's I'd say is very unique about the Flagship Pioneering model, is this mindset about really asking the tough what if questions and seeing the world in very different ways that perhaps I feel like I've had the opportunity to explore during my almost 20 years in big pharma. It's very much about like thinking outside of the box, and instead of, maybe being thought of as somebody who was a little bit weird for asking really tough questions about, well, why do we do it this way, which was a little bit my experience in pharma. It's encouraged here to really challenge the status quo, really think big and broad. And that's really the genesis of these tremendous ideas. It's not about the adjacencies of ideas that we already have. It's about making bigger leaps towards innovation at a much different pace than perhaps we've been able to do before in the past.
Yeah, and thinking along those lines, if you think about the traditional pharma sector, they've been historically sort of avoiding areas where there's a low population count. So rare disease spaces, for example, primarily because I think there's just not enough of a market for them to grow or develop that sort of those sorts of drugs. So I know that you're in the genetic space here. So what sort of are the challenges for R&D that are targeting genetic
Yeah,
diseases?
what I think is so important, and you bring it up specifically about one of the challenges, especially from a pharma perspective, is, when you think about the world of genetic diseases, there are at least 6,000 of these different conditions. Actually, if depending on who you ask, they might say it's about 10,000 different diseases. It really depends on how you count and who you ask. And because some of these are not as well characterized as others, it's really hard to get a handle on the exact number. But we're talking about a lot of diseases, thousands and thousands of these. And, we've got some very exciting modalities that are, certainly emerging today. But the reality is the way that we're trying to tackle this number of different diseases is using a very conventional approach. And that's using a very disease by disease or gene by gene type strategy. And when you have 6,000 or 10,000 of these different conditions, you can imagine the numbers of lifetimes that it would take, Ray, for you and I, in order for us to really be able to make a dent in that. And that's predominantly one of the reasons why, when in the exploration that's being done in the rare genetic disease setting, the focus has been on the few that at least have a certain, numbers of patients that are meaningful enough. But that's also one of the reasons why there have been few innovations in this space. And when we look across the rare genetic disease setting, there are, only about 5% of these have an FDA approved medicine today. So the innovation within this space is actually quite low. And so the unmet need is actually tremendous. And so across the rare disease setting, but then also the genetically driven cancers, of which there are several, and there are no approved medicines that are aimed at correcting those mutations, this leaves actually a tremendous population that is underserved by modern technology as we know it today. And by our best guess, we think that there's around 300 million people living around the world with these genetic diseases that are, being underserved as we know it. And so this is precisely the patient population that we're looking to tackle with our novel approach and our novel look at the world here at Alltrna.
So tell us about how you founded, or you are leading Alltrna, and sort of what makes Alltrna different or special. What's the magic sauce there?
So the whole premise behind what we're doing, so first of all, we're leveraging the biology of tRNA, transfer RNA, which we'll get to in a moment. But the real interesting thing around what we're doing here at Alltrna is thinking differently about how we tackle the world and how we classify patients, where we may have the opportunity to have a single medicine that can be used to treat hundreds, if not thousands, of these genetic diseases. So we talk about, taking this disease by disease approach, but can you imagine a world where instead we may be able to simplify that problem into a number of universal diseases that then can be tackled in a much more streamlined way, where we can develop medicines for that approach and then hopefully be able to treat patients with a different approach. So when we think about patients with genetic disease, we reclassify them differently. So if you actually pull up our next slide, Ray, I can sort of talk through that. It's slide 3 in the deck that you have. So right now, patients are today classified using this conventional means, disease by disease by disease, and and those diseases are generally grouped by their clinical presentation or the gene that's affected, and that's why we have over 6,000 of these different conditions. But we think very differently about how we reclassify patients with these genetic diseases, because what we do is we look for the underlying mutations that are common across all of these different diseases. And we observe that the same mutations can be found in each of these different diseases, which is actually quite important. And by bringing those patients then that have those shared mutations, so instead of a focus on the disease, but by the genetic driver or that specific mutation instead, this actually opens us up this whole new world of possibility for us to be able to treat these genetic alterations and to use so with a single medicine that is specifically designed to treat that mutation. Seems like it could be a novel concept, but I will share with you, this is actually an approach that we've seen have success in other settings, such as oncology, which is where I've spent most of my career. And if we take that same sort of playbook that we've had in the oncology space and apply it here in rare diseases, I think this really could be extraordinarily transformational.
It sounds very interesting. How long has this sort of technology been available, and how long have people been trying to do this?
So maybe let's get into a little bit about the basics of tRNA first, and then we can kind of come back to that, which I think would be helpful. So the tRNA is, what we find is a universal tool in this protein translation process. So when you think about the genetic diseases that we're talking about, most of them are caused because of a monogenic, there's a mutation and there's a protein function that is disrupted, or the protein isn't formed correctly, and that's really what causes that disease. But what was misunderstood or not well understood before is really the importance of the tRNA, the transfer RNA, as having this universal role in this protein translation process. And it's really that understanding of the biology that has really inspired the work that we're doing here at Alltrna. So if you just press play on this video, I can talk you through that a little bit. So here we're looking at zooming into the protein translation infrastructure inside the cell. So you see the strand of the mRNA, it's surrounded by the ribosome, which is that machinery. You see this colorful growing polypeptide chain, protein chain on the left-hand side. That's exactly right. But if you look inside the ribosome, what you see in that sort of unique L-shaped structure there, that's the tRNA, the transfer RNA. And its role is to specifically read the code of the mRNA and understand the precise instructions, and then to know which amino acid is being coded for there, and then to transfer that important amino acid to that growing polypeptide chain, which is exactly what's needed for biological function. And when you look at this sort of scene here that's on the slide right now, you can see that it's this lens here, that the tRNA is actually the physical link between the mRNA and the protein. So it really speaks to the criticality of its specific role. And really, without tRNA, there would be no protein. That's how important it is. What's also exciting is that the tRNA is actually a truly universal tool and a truly universal role in this entire process. It does the same job no matter what gene, no matter what mRNA strand, no matter which protein. It's always the same job over and over and over again. So unlike other modalities, maybe that require a specific protein or a specific mRNA or tackle a specific gene, the tRNA is always exactly the same in the role that it does, which is, what makes it truly special.
Interesting. So, check if there's another slide that you want to go through
Sure.
here, or
Well, let's, now come to a little bit about, well, how long have we been doing this and where are we starting? Because I think that's actually, a good segue to, the next part of the discussion, which is, so Alltrna has been, intrigued by this role of tRNA for about 5 years now, really since 2017. We started to really dig into this. In fact, the sort of founding team, and that's when we started to really put our heads together, not myself, but predecessors before me, other people who are still involved with the company. We started to really explore and try to understand, well, what if you can alter the protein translation process by altering the way in which the tRNA does its role? And that has really inspired the work that we're doing at Alltrna. It really, people understood what the tRNA function was, but didn't really understand how to unlock it into a new modality. I'd say in part because we didn't have the tools to really understand the sort of basic principles of a tRNA, how to measure it, how to understand, where, what its characteristics were, where it was going. All of those things had to start. And that was some of the foundational work that we had to do. But where we're beginning is actually looking at a specific type of mutations that actually occurs quite commonly in the world of genetic diseases. So we're starting with what are called premature termination codons, or nonsense mutations, which actually occur in about 10% of all genetically driven diseases. So when we think about the sort of, 300 million people that I've mentioned that have those genetic diseases, the patients or the people that would have a disease that's caused by one of these premature stop codons is about 10% of those. So roughly 30 million people living around the world that have a disease that's caused by one of these types of mutations. So it's actually quite substantial. Now, what we actually call this group of patients is, it's having stop codon disease, a very different way of thinking about the condition that they have, not by the sort of classical definition of a disease, but actually by having stop codon disease, because we're looking at the genesis or the foundation of that, mutation that is specifically causing that disease. But to understand that more, maybe let's go to the next video on the next slide, Ray, and I can talk a little bit about, well, what exactly is a premature termination codon and how does it work? What it is that we're doing here. So
all right,
that's here on this slide, which is, we're all about reading through these premature termination codons to restore protein production. So you see on this slide, this little red, codon here, that's a premature termination codon. So what happens in nature today is that there are no tRNAs that can read those premature termination codons. So what happens is the translation process ends too quickly. The result is a truncated or shortened protein, and that's what causes disease. Now, what we're doing at Alltrna is actually engineering tRNAs that understand how to read through these premature termination codons. And by doing so, that translation process is continued as normal. So it reads that codon, it understands what amino acid is next, transfers that correct amino acid, and the translation process continues all the way into the end when you expect it to end, and it restores that full-length functional protein, which is precisely what patients need in order to address that disease. Now, what's also truly exciting is that we see the same premature termination codons from disease to disease to disease. So that introduces this element of universality, of us being able to use the same engineered tRNA in order to address all those numbers of different diseases. If you actually go to the next slide, Ray, this gives another sort of picture using stills, which may be, an easier representation or another representation to get this concept. So you can see here on the left-hand side in that healthy state. So in most people who don't have these types of mutations, you see that translation process continues all the way until you have a full-length protein, which is, precisely what we need for our normal sort of cellular functions that make up life as we know it. But oftentimes, and as I mentioned, in 10% of cases of genetically driven diseases, there's a premature termination codon that exists. This is when, the code for an amino acid is mutated, and instead it codes for a stop, which means that the translation process stops precisely at that premature termination codon. You can see that where that UGA is in red. And that result is that truncated protein, which is what causes disease. Now you can see here on the rightmost panel is precisely what we're doing here at Alltrna. It's engineering this, very elegant structure, which is this more or less 76 nucleotide, base, oligonucleotide, which then can read through, specifically designed to read through these premature termination codons, so that that whole translation process continues. And again, that result is that full-length protein, which is precisely what patients need.
How do you deliver the engineered tRNA into the body?
Yeah. So with all oligonucleotides, it's very much dependent on how you formulate these to make sure that you can get to the tissues and the cells that are of interest for all these different diseases. Now, we are looking at, in our first instance, about formulating with a lipid nanoparticle, which means that we're going to be targeting liver diseases, so rare genetic liver diseases in that first instance. We're doing that specifically, and I'd say it's a strategic choice to do it that way, because we want to make sure that we can take delivery risk off the table with a brand new modality like a tRNA that's never been tested in humans before, so that we can focus on the tRNA and make sure that we can fully understand the safety and benefit that payload is providing with a clinically validated delivery mechanism that is well characterized and well understood.
Right. And so since this is like a first of its kind tRNA technology, are there other companies or even labs trying to do this like you are, or are you significantly ahead of the curve?
So what I would say is, until recently, there's been very little focus on the tRNA space. And certainly within the last couple of years, we're starting to see that there's a little bit more attention that's being paid here. I actually find this to be a very encouraging sign, because I think people are starting to realize that maybe this is an area that was overlooked. And now perhaps with the learnings that we've had from mRNAs, with the learnings that we've had in other spaces like siRNAs, that maybe now is a more optimal time to really unlock its potential to become a new modality. I think that's encouraging news. I will say that we've been at this now for over 5 years, and we've certainly have made a tremendous amount of progress. I don't think anybody is really looking at it in a truly sort of platform way in which we are looking at the tRNA biology. We're talking specifically about engineered tRNAs and premature termination codon mutations today, but I think that's just the tip of the iceberg, and Alltrna is already starting to explore other applications of tRNA that may also be very beneficial for patients across a broad set of different conditions, which we're excited to explore in the future.
Wow, that's very interesting. And I'm sure engineering these tRNA molecules is really challenging. Are you leveraging any sort of artificial intelligence or massive amounts of data in order to generate the right shapes and the right amino acids to deliver what you want?
Absolutely. And in fact, it's one of the things that I think really truly sets Alltrna apart from others who are exploring this area. For sure, when it comes to our platform, which is all about how we design our tRNAs and then how we characterize them, it really has an underpinning of our platform that is very digital and machine learning based. And let me explain to you why that's really important. As I mentioned, that the structure of a tRNA is a 76 nucleotide structure. It's, before we started the work that we're doing here at Alltrna, actually nobody had chemically synthesized a 76 nucleotide structure before in the past. So that's something that we had to really demonstrate that we can do. But we've also learned that how you sequence those nucleotides play an important role. And then how you decorate or chemically modify those nucleotides is also equally as important. And so through sequence optimization and through modification optimization, we've actually learned that not all tRNAs are created equal. In fact, not all engineered tRNAs are created equal, and that we can actually design for really important clinical features such as stability and selectivity and potency and immune profile, for example. These can all be designed for by using those different optimizations. Now, the numbers of optimizations that are possible is incredibly vast. In fact, between the sequences and the modifications, there are combinations that are like the numbers of combinations are greater than the numbers of atoms in the universe. That's a lot, right? That's a lot.
It's too many for a human to write down on a
The
piece of
most
paper.
brilliant Nobel Prize winning humans would not be able to make the best selections on their own. And so it really requires this engine in order to be able to help enable our researchers, our scientists from being able to have the predictive power to select those designed tRNAs that have the best likelihood of having the clinical impact that we're looking for. And our data really demonstrates that that's exactly true. We find that our engineered tRNAs that are selected using this machine learning tool are actually far outperforming those that are explored using a more rational, conventional approach.
Wow, thanks for sharing that. As you mentioned before, there are over 300 million people with rare disease, and then 10% of those have this premature stop codon, which is actually causing the disease. So that's specific mutation in one place in their genetics. So can you reiterate what is the significance of that, and what are really the opportunities here? How significant is this, and when can we see sort of this in clinical trials as well as commercially used?
Yeah. I mean, so not only are we talking about a huge number of patients, but the way that I think about it is like the opportunity to really bring together and simplify the way that we develop these medicines and perhaps even treat these patients can be extremely substantial. So as we've talked about before, conventional approaches, disease by disease by disease, gene by gene by gene, that's how drug development for rare genetic disease is being done today. And that means that there's going to be, I don't know, 5, 10 of these different rare genetic diseases that are going to get 90+% of the attention. Now, that means everybody else gets none, right? And the reason that, one of the things that truly excites us is the opportunity to have a massive impact on patients. Yes, of course, not only those patient populations where there are a sizable number of patients, but also because by looking at these same mutations across all these number of different diseases, it doesn't really matter so much to us that there may be thousands in one disease, but then only hundreds in others, because we can bring all these patients together. And we think actually there's an opportunity to create a development plan, again, using the oncology setting as a model where these basket trials have been very successful in leading to FDA-approved medicines before in the past, including a couple of programs that I've had the opportunity to work on. But this may actually mean that patients who have certain diagnoses, who right now would do that clinicaltrials.gov search like I did, and might find that absolutely nothing is being done in that disease, that may not be true anymore. We may be able to capture many of those patients in addition to those few diseases that are getting some of that attention into a single, unified, simplified clinical trial plan. That means faster, maybe, exploration or innovation for a number of these different diseases, that especially those that aren't getting attention today. It also means less of an investment that's required from an R&D perspective, because we're able to unify the development process in such a way that would not mean that we'd have to do clinical trials for each and every one of these different diseases. And when you have efficiencies from a time and financial perspective, I think this is exactly where patients have the opportunity to benefit. So I'm really excited about that. And so I hope that we can, work towards a world where, if you're in an unfortunate circumstance where you or a loved one gets diagnosed with one of these conditions, instead of, doing that research and being like, wait a second, there's absolutely nothing for me, you might actually get to a place where it says, oh, okay, there's something on the horizon. It actually is quite, somebody's paying attention to this, and it actually could be promising. And that gives a lot of sort of hope and optimism for what the future can hold.
Yeah, I mean, this would be a massive breakthrough, and it would be cool to see, you know, even hundreds of clinical trials with the same drug for 100, maybe 200 different diseases, maybe thousands of these diseases. So very interesting, very exciting too. So thanks for sharing that. Changing gears a little bit, moving into the fundraising and biotech landscape sort of space here. So how are you seeing the biotech funding landscape? And has it affected the way you're seeking your next round of funding?
Yeah. So I think it's interesting. I think when you look at like a snapshot of where we are today, and especially if you maybe are comparing the funding landscape to where the industry has been over the last 2 to 3 years, you might say like times are tough right now. It's different. It's, harder than it used to be. I would actually argue in some ways that, the last 2 or 3 years were maybe perhaps the anomaly. And we've now had a little bit more of a normalization of fundraising within the biotech sector. So to me, what I would say is the recipe for fundraising, I think, is essentially the same. You've got to be able to create a compelling argument about what the unmet medical need is. You've got to be able to create a compelling story about how your technology has that opportunity to in a very differentiated and transformational way, be able to address that unmet medical need. And you need to be able to demonstrate that you've got a team and a plan that can execute on that strategy and really be able to bring it to life in a way that makes, I'd say, good use of the resources that we have afforded to us. And I would say that for me has certainly been, the way that I'm looking at it. And when I talk about the Alltrna story, which, we've had the opportunity, Ray, to speak a little bit about today, I think that it's quite compelling, to be able to think that a single medicine can be used to treat hundreds, if not thousands of different diseases is something that really no other novel modality today can say. And thinking about the numbers of patients or the numbers of different diseases that can be tackled using a simplified approach is certainly something that, I think is compelling for lots of different stakeholders.
Yeah, absolutely. I think, it's interesting you mentioned how during COVID, it was sort of like the anomaly. People were speculating a lot. And I feel like now we're going back to fundamentals with how investors are looking at different companies and choosing which teams, which science makes the most sense to them. So in a way, it's good.
It is, right? And I think no matter what, we're always going to have to work hard to bring new medicines, forward, right? It's always going to be the case.
It's not an easy process. Absolutely. What is Alltrna's next inflection point? When you think about inflection points in terms of what study or specific, milestone are you trying to achieve that will help you sort of increase your valuation significantly, for example?
So as I mentioned, we've been sort of working behind the scenes for the last 5 years and made tremendous progress on the platform as it stands today. And we are really at this very exciting juncture for Alltrna, where now that our platform, we've made so much progress, we are at that critical time where we may be transitioning from a, platform-based company. Well, we'll always be a platform-based company, but a platform that may also have development candidates that can turn into pipeline assets. I mean, we have this ambition to really make an impact on patients. The way that we do that is by entering into the clinic and being able to demonstrate proof of concept. So for us, it's really about working towards that major milestone. And that's where we think that we're really going to be able to demonstrate the tremendous potential that tRNA biology has and the platform that we've put in place in order to be able to deliver it.
That's great. What is the progress so far in achieving?
Well, we've made a lot, right? So we've already have really encouraging in vitro data. We already have really encouraging in vivo data that exists today. In fact, we've been able to demonstrate that our engineered tRNAs have the ability to read through. We've tested it in over 25 different disease models across 14 different genes where mutations exist in different locations on that gene. And each and every time we're able to show that we can read through and rescue protein, just like we've talked about in those videos. So for us, I'd say we have, certainly managed to build the company and make progress scientifically. And now we'll be sort of like pulling all that together and starting to work towards a plan that will hopefully be able to get us into the clinic into the not too distant future.
Michelle, thank you so much for sharing all that, and I appreciate your time today. This is my final question, and then we can, kind of close out here. What is the most exciting thing that's happening the rest of the year for Alltrna?
Every day around here is an exciting day. But when I think about what makes Alltrna so special, we've talked a lot about the science today, and that is truly, truly unique, as well as our approach. But at the core of everything we do are the people in the team. And we're making tremendous progress in continuing to build out the Alltrna team, continuing to invest in the culture. There's going to be a number of critical appointments, especially at the C-suite, that are really going to help us with this transformation as we, carry on this journey. And so for me, it's really all about that, right? I'm most excited about working with the people and building that scientific and leadership expertise that is really going to help us at this pivotal next stage. And that to me is the most exciting and most important thing that's on the near-term horizon.
Fantastic. Thank you so much for joining me. And if you're interested in Alltrna or tRNA technology, or you'd like to reach out to Michelle, please feel free to find the show notes to be very helpful for you. So just check those out. There's links to the website as well as your LinkedIn. And is there anything else you'd like to share with the audience today, Michelle, before we conclude?
I just will leave with this thought, in that we've got this real opportunity on our hands to fundamentally change the way that we think about rare diseases. In fact, in some ways, when we group patients in this different way, classify them by their common mutations, maybe they're not so rare anymore. And so maybe perhaps some of those sort of like common issues that were roadblocks previously in the past in terms of bringing innovation forward could actually be real opportunities. And that to me is, what I think is truly, truly exciting. And I say that both as the CEO, but also as a mom, right? To fundamentally think that we might not be in this world where, in the rare space, it can feel very isolating and alone, but maybe there's a lot more of us than we think that there are. And, coming together in this way and thinking differently about how we see this community, I think could be really, truly groundbreaking.
That would be an amazing result, and I'm looking forward to that as well. So thank you again. And if you're interested, make sure you subscribe, follow, like, share, and catch us next time.